First presentation for today will be given by Dr. Fekade Sime, who will then assume the role of being the moderator for the rest of tonight's session. Fekade is one of our shining stars in CRE Respond. He's an NHMRC Emerging Leadership Fellow and is based here at the University of Queensland Centre for Clinical Research. His main funded project looks specifically at trying to characterise the stability of antimicrobials and their appropriateness for use in outpatient parenteral antimicrobial therapy scenarios. And he has very extensive expertise in preclinical infection models and also pharmacokinetic and pharmacodynamic modelling. Welcome, Fekade, and please take it away.
Thank you, Jason, for the kind introduction and also for setting up the scene for this good opportunity. My talk today will be on antimicrobial stability studies in OPAT. I will mainly focus on highlighting some of the challenges in hot tropical environments. So the outline of my presentation will be a brief background to begin with, and I will highlight the challenges in the terms of temperature conditions and environmental exposures for OPAT infusion devices in tropical settings. And next, I will give a bird's eye view of available stability studies for the major antibiotics of interest in OPAT. And finally, briefly describe the potential role of PKPD principles to apply in the OPAT stability study settings. So the definition of OPAT as given by IDSA, it refers to the administration of intravenous antibiotics in at least 2 different occasions without the intervening hospitalization. So OPAT is currently administered in different modalities, mainly 3 modalities, either in the home setting within the community or infusion centers within the community. And in some jurisdictions, there are specialized nursing facilities that provide administration of OPAT within the community. The most important concern is mainly in home-based OPAT because healthcare professionals do not have a direct control in terms of the handling of the devices and exposure to environmental conditions. Especially in the home-based OPAT, there can be 2 modalities of either self-administered OPAT or healthcare professional visited administration, especially in self-administered OPAT, which is often referred to as OPAT. So we don't have, as healthcare professionals, a direct control on the handling of the administrative devices. So especially when the mode of administration in OPAT is a continuous infusion mode for antibiotics that are usually given in multiple daily doses in a hospital setting, exposure of the antibiotic to environmental conditions for extended period of time is a serious concern in terms of stability of the drugs. So because of this reason, and stability of antimicrobials has become one of the primary selection criteria for antibiotic use in the OPAT settings, in addition to the most important clinical criteria such as efficacy and dosing frequency or suitability, etc. Why is stability a concern? So stability is a great concern mainly because of the high temperature exposures to infusion devices and the solution within the infusion devices. So a typical study, a very interesting study published in the Journal of Antimicrobial Chemotherapy in 2017 from a group in Switzerland, investigated the exposure to various temperatures through different seasons. And they basically looked at temperature exposures when the devices were carried around the waist, which is the solid line that you see on the slides, or when it is carried by the patients but a bit away from the body and besides the bed during the evening. The shaded area in the middle of the graph that you see is the data for the evening, but you can clearly see how the temperature rises and goes beyond 25 degrees Celsius, which is the normal room temperature, and plateaus at a temperature a little bit above 30, and especially when the devices are carried around the waist. So The mean temperature when carried around the waist was around 31 degrees Celsius, but when kept besides the bedside, especially during the evening, can be slightly lower. And a direct exposure to the sun in the summer can significantly spike the temperature exposures. So up to 38 degrees Celsius, for example, with white pouches or carrying pouches, or up to 45 degrees Celsius in black pouches. So temperatures can actually go beyond the usual temperature to a high level above 30 degrees Celsius. Another interesting study from Australia, from a subtropical climate area, is a study published in American Journal of Health System Pharmacy in 2017. Again, this study also looked at a 12-month period temperature exposure to infusion devices. This is a study during routine clinical use. It was one of the first studies to record temperatures over multiple seasons during a year period of time. So we enrolled about 115 patients with more than 30,000 temperature recordings from the devices. So they attached the temperature monitoring device on the infusion pumps, and patients were recordings were collected for different seasons. So interestingly, this study also included the infusion location time during daytime, also and during evening times, as you can see in the table on the right. So most of the patients carry the devices on their body during the daytime, and a substantial proportion, around 20% of them, also carry the devices on their body or within the bed during the evening, which is a major reason for a spike in temperature, as you have seen in the graphs in the earlier study. So this study reported a wide range of temperature readings from the data loggers. So temperature range were from 9 degrees Celsius up to 45. The median temperature was 26 degrees Celsius, however, but there were substantial proportion of the recordings above 25 degrees Celsius. So 60% of the readings were above 25 degrees centigrade, which is normally considered as a standard room temperature in laboratory experimental settings. Also, up to 70% of the readings were above 30 degrees Celsius, which is a bit of a concern because not so many antibiotics are stable over prolonged exposure at these high temperatures. So they have also seen temperature exposures stratified by different seasons. Obviously, as we can expect, in the summer, the top curve temperatures are high compared to the winter, the blue in the bottom, and for spring and autumn, so comparable temperature readings. Most interesting from their data is, so the median percentage of readings above 30 degrees Celsius and 25 degrees Celsius, especially during the summer, were very high. About 95% of the readings during the summer are above 25 degrees Celsius, and about 20% of the readings are above 30 degrees Celsius. So substantial proportion of the infusion devices get temperature exposures above the standard 25 degrees centigrade room temperature. The location of the devices overnight has also a significant impact. So when they are kept on the body or within the bed overnight, so the temperature remains high, just like as in the day, compared to keeping the devices away from the body or from the bed. One limitation of the previous study was the data loggers were kept externally on the devices. But this interesting study, again from tropical climate in Australia, investigated the actual temperature of the solution within the devices in Townsville in Queensland and in Darwin, Northern Territory. So the maximum temperature they recorded within the solution was 34 degrees Celsius. So the authors also reported the comparative results when patients have access to air conditioning versus when they don't have access to air conditioning. Using a linear mixed field modeling, so they reported that for non-air conditioned exposure, the temperatures are slightly higher, and the mean temperatures were around 32 degrees centigrade, compared to the air-conditioned environment where it's slightly lower, up to 25 degrees Celsius. When we look at the 90th percentile of the temperature of the solutions for non-air conditioned environment, it is 33 degrees Celsius, 1 degree above the mean, and for air conditioned is nearly 32. So all data analyzed together, the 90th percentile of the temperature of the solution is 32.5 degrees Celsius. So in general, it looks like the temperature of the solution mostly is around 32 degrees Celsius, or within 1 degree Celsius of the 32 degree centigrade. So this is reflected in the standard regulatory documents that's prescribed. So the protocols for stability testings. One of the documents, which perhaps later Dr. Jamieson will describe in detail, is the UK NHS yellow cover document that you see on the right side here. It is a standard protocol for delivering assessment of stability studies for small molecule aseptic preparations. For OPAT specific temperature testing, this guideline recommends 32 degrees centigrade plus or minus 1 degree centigrade as an in-use temperature to test. And this is justified by most of the data that I've described later, and also other similar studies. So this document is also prescribed additional critical points that stability studies should consider, and which most of the traditional stability studies do not account for. Based on the information available in terms of exposure temperature and also the regulatory requirements of the yellow cover document, which we have seen recently, there was an interesting systematic review published from the Drug Stability Working Group of PSAC in 2017. So they looked at all stability studies for OPAT preparations that meet regulatory requirements based on the then yellow cover document requirements, which also included a more stringent testing at 37 degrees Celsius, which was later amended up to 32 degrees Celsius in 2019, I think. So in general, they found that none of the published studies prior in that period meet the regulatory requirements for stability studies. Even when considering those which tested at 37 degrees Celsius, they did not meet requirements for stability testing on other criteria. One of the recent systematic review study that summarizes the stability of antimicrobials infusion devices was published this year on the journal Antibiotics. So they included all studies on antimicrobials, including antibiotics, antivirals, and antifungus, which aim to evaluate the physicochemical stability of antimicrobials stored in infusion devices. And this systematic review summarized data available on 1st of July 2020. They were able to find in their systematic review from over 600 articles they screened 33 studies, and in their qualitative analysis, they reported that most studied antibiotics in infusion devices for stability are the beta-lactams. This is reasonable, as we all know that the beta-lactams are highly thermolabile. And among the beta-lactams, the penicillins are the ones which are most studied, amoxicillin, flucloxacillin, and piperacillin tazobactam being the most frequently studied, and Kefazulin and Kefazulin from the cephalosporins, and meropenem from the carbapenems. So what I would like to do now is just to give you a brief overview, a bird's eye view of available stability data and whether they can really really comply with existing requirements in terms of the temperature exposures to inform clinical decision. If you look at the available studies on the stability of penicillins, for ampicillin, we've got a couple of studies available, but as you can see from these tables, one of the studies did not include testing at 32 degrees Celsius, which is any use temperature, but the other one did include 31 degrees Celsius. However, still, it did not study other factors, including physical stability, and no stability was reported. This review article summarized acceptability in terms of chemical stability based on less than 10% degradation. So ampicillin was not stable. So the next antibiotic, amoxicillin. So there are 4 studies. None of these included 32 degrees Celsius. We recently done in collaboration with the British Society of Antimicrobial Chemotherapy, a periscoping study at 32, and we found that amoxicillin degrades significantly in a concentration-dependent manner, and none of the available studies also support its stability at 32 degrees Celsius. For benzyl penicillin, there is one study which does not include 32 degrees Celsius as in-use temperature, so not that useful data. So next, for flucloxacillin, there are 4 studies. Perhaps the most important study to highlight is the one again from the BSAC drug stability working group. This study is really an interesting study with a high quality in terms of the design. It includes all the features prescribed by the contemporary regulatory guidelines. And this study has demonstrated that when buffered in 0.3% citrate, flucloxacillin is stable, meeting all the regulatory requirements with less than 5% degradation, even at in-use temperatures as high as 32 degrees Celsius. But for the rest of the penicillins on this slide, so there is no regulatory compliance study for meslocillin, nafacillin, penicillin G. For piperacillin tazobactam, on the study from the BSAC drug stability working group is the only one that complies with all the regulatory testing requirements, including high temperature exposure. And piperacillin tazobactam was also demonstrated by this study to be stable for 24-hour infusion. The next antibiotic, temocillins. So there is one published study, however, it does not meet regulatory requirement. Again, in our ongoing collaborative work with the BSAC drug stability working groups, so we have recently done a stability study on temocillin, and we were able to show it can be stable when in 0.3% citrate buffer if we are considering less than 10% degradation for 24 hours, or in jurisdictions where less than 5% degradation is required, just like as in the UK, it can be administered as 12-hour infusion twice daily. Kefazulin, for Kefazulin, there are multiple studies, and as you can see in the middle, temperature and duration of storage columns, so the temperatures tested do not replicate that of the OPAT condition. So there is one study that complies with the requirements, which I will highlight later in another slide. For the other cephalosporins, again, ceftazidime is the one which has got a study that complies with all regulatory requirements. No good data is available for the rest. Again, this comes from the BSAC drug stability working group for ceftazidime. Ceftazidime can be administered as a 12-hour infusion because it was not found to be stable for 24 hours. It was stable up to 18 hours when reconstituted in 0.9% saline. Also, because ceftazidime is one of the antibiotics where degradation products are a big concern, specifically pyridine. And this study highlighted that only half of the volume of the infusion can be administered without exceeding the pharmacopoeia limit for pyridine, which is 0.5% weight by weight. So for these 2 reasons, ceftazidime at the moment can only be administered 12 hourly. For the other Kefazulin, antibiotic, which is Kefazulin, Tazobactam. We have another high quality study from the BSAC working group, which demonstrated again stability of over 12 hours when we consider less than 5% degradation. So we did a study in our group as well, including 37 degrees centigrade previously, and we demonstrated that it is stable if we consider less than 10% degradation over 24 hours. But for stringent requirements of less than 5% degradation, this antibiotic can be given again only over 12-hour infusion. For the carbapenem antibiotics, so this slide summarizes available studies, but to cut it short, so the temperature conditions tested do not comply with the OPAT-like exposures. So generally, there is no study at the moment that complies with all regulatory requirements and demonstrates stability of the beta-lactams for OPAT use and of the carbapenems for OPAT use. Sorry. Next slide, please. Next slide, please. So for other antibiotics, again, for the data does not comply with regulatory requirements for clindamycin, colistin, and colistimate. Next, for antifungals, only 2 antifungals have been studied so far from the published literature. And for the antivirals, only ganciclovir published study is available. We're currently studying acyclovir again in collaboration with the BSAC working group. So in general, so what I wanted to highlight is the lack of stability studies that comply with regulatory standards, although some of these antibiotics are also used in practice. So this highlights the need for further testing, considering all the standard criteria that needs to be included in the study design. Another recent latest review has from the BSAC working group also highlights that although previous studies were not meeting the regulatory guidelines, but in recent years, since their first publication of the review in 2017, so few of the emerging studies do have a high quality, and they are considering all of the regulatory guidelines in the yellow cover documents. I've highlighted some of these studies in the antibiotics that in previous slides. So to conclude on the future directions in the stability testing could be to integrate the principles of PKPD and specifically PKPD modeling to integrate data from clinical studies and stability studies and through modeling and simulation, and predict optimal dosing regimens for those antibiotics which may not meet the stability criteria set forth in the guidelines or which marginally meet the guidelines. So when the safety of course the degradation product is acceptable. So perhaps this is a future direction for this research, maybe for discussion, but eventual approach to optimize the dosing regimens when marginally stability is observed from the stability studies. So with this, I would like to conclude my presentation.